Okay, so next up we have Steve Kelly. So Steve's 19 years old. He comes to us from WPI, where he's just starting his junior year in mathematics. He'll be telling you about inkjet printing of genetically modified living bacteria.
Okay, thanks Jordan. So hi, my name is Steve Kelly, and I'm here to present inkjet printing of genetically modified living bacteria. So I guess I'll just start with explaining what my interest in making is. So I really got started in high school with building robotics, or robots, and I experienced that through our first team, which stands for Inspiration and Recognition of Science and Technology. So at my high school, we only really had access to a wood shop. So I was always thinking about how I could potentially make more complex things. And that's when I discovered 3D printing. And ever since then, I've pretty much been making my own tools at both the software and hardware level. So I'm a mathematics student, but I like to say I'm an engineer at heart. And at AMRI, my real interest has been in learning more about scientific analysis and biology. So we had some inspiration for this project, and that happened in January. So BioCurious, their community biology lab in the Bay Area, posted this online. So essentially it's a 2-axis motion system. They took apart some CD players and attached an inkjet head to it, and they were able to print bacteria. So it's biofluorescent. But we saw this and we thought there was a few things we can improve on. And one of those was accuracy. As you can see, there's a lot of overspray here. And we were thinking about what happens if we add multiple types of bacteria. There's some interesting things we could look at, such as mineralization, pigment formation, and bacterial films. So we thought about this and realized we need something that's accurate on boundaries and precise. And we're also looking at CD players, and we're thinking, okay, it should be easier for people to build. You know, taking apart CD players isn't exactly the easiest thing to do. So our proposal was to develop some mechanics and patterning systems, and in my case it was to learn about bacteria. So from science, you generate theory, and from theory, you generate engineering. And we wanted to combine those two and take a look at what kind of tools we could use to help with scientific investigation. So one of the first things we looked at was how thermal inkjet heads work. And we wrote about the history, and the inspiration was pretty interesting. So it was an engineer took a hot iron on a syringe, and when he did that, he realized that some of the liquid was expelled from it. So what they did was they miniaturized this, and as you can see in the picture on the right, there's a heater at the top which generates a bubble, and it just expels a little bit of ink. It turns out bacteria can survive this. It's not too hot, and there's a big buffer. Unfortunately, human cells are too big to go through an inkjet head. So next we started looking at control algorithms. So how do existing 2D printers work? So we're wondering if we could just put bacteria into a regular 2D printer, what would happen? So it turns out that regular 2D printers work by having cyan, magenta, yellow, and black. And they mix all these two. And they do it by angling the patterns. And they overlap. So we wanted discrete boundaries. So we realized this wouldn't work. So by having them overlap, it creates this, it tricks the eye into seeing gradients of color. So next we started looking at the geometry of bacteria. How do bacteria grow, right? And it turns out when you place them down onto a surface that has nutrients, they're going to expand, they're going to multiply, and they multiply, and they tend to create very nice circles to minimize surface area. So what we realized was we weren't so worried about creating a continuous picture as much as creating accurate and precise placements. So at this point we kind of have a picture of what we should build. So one of the things we were thinking about is when we were looking at that picture, we were calling all the inaccurate placements overspray, right? Which reminded us of a spray can, right? So as you go farther from the spray can, it becomes more inaccurate. So one of our first hypotheses and experiments was that to improve accuracy, we need to control the distance. So we started looking at different types of hardware to enable this. And we looked at what the biocurious people used, and they had a very good inkjet system called Ink Shield. And as you can see, there's no way to control it, right? In this example, he just moved it by hand. Next, we saw the rep wrap, as Jordan mentioned, and that gave us a easy-to-use 3D printing system. And it turns out that both these use the same control system, so we're able to merge the two together nicely. So we needed 3-axis motion in the case of the rep wrap for our first experiment, which was adjusting height. So this is the first experiment we did, and that was to quantify accuracy. So we printed a bunch of strips at different heights at the top, and we developed some image processing software to quantify the shape of the droplets, the size, and the accuracy, and also the number. So indeed, what we found out was that we need to stay within 0.5 millimeter of the surface to be really accurate. So next we started looking at creating some path creation software. So we wanted some way to create those discrete boundaries, and of course existing 2D printing is not going to work. So what we looked at was creating a basic program that converts images to control toolpaths that we can send to the rep wrap, that then we can output and or send to the printer and generate output. So one of the next things we thought about was doing surface experiments. What kind of effects we would have when we printed on different surfaces. So here you can see when we just print regular ink onto agar, which is the growth medium for cells, it kind of diffused. So that was taken about 30 seconds after it came off the printer, and it was nice and sharp, like you see back here. But within 30 seconds, it was like that, and then within a minute, it was like that. So it really kind of just gets hazy, and that was because of some of the liquid on the surface. So we started experimenting with printing on the paper, and then transferring the bacteria, or printing bacteria onto paper, and then transferring the paper onto the print surface. So we started looking at that, and we realized that some of the liquid was spreading across the surface. And that when we removed some of that liquid, we were able to get accurate deposition. You see in the bottom right. So at this point, we haven't gotten to printing bacteria directly onto the paper with the machine. We're doing it by hand. So that's something BioCurious has already done, and we just need to add that in. So we've quantified and qualified our operation, and we've created some sort of framework. So the next step is to document thoroughly to allow scientists and hackers to expand on this and experiment. So I'll finish by saying why AMRI was such a great experience, and I think it's kind of summed up in this quote by Richard Hamming, who was a researcher at Bell Labs. So he says, in science, if you know what you're doing, you should not be doing it. In engineering, if you do not know what you're doing, you should not be doing it. Of course, you seldom if ever see either pure state. So AMRI kind of merges both of those two and creates that kind of perfect environment where you can do science and you can do engineering in a structured manner and see good results. So my project's documented on GitHub in a repository. And does anybody have any questions?
Why would you want to print bacteria?
So the question was, why would you want to print bacteria? So there's a lot of incentive for that. And we were looking at some of the papers. I was starting to understand some of them about how you can create minerals on boundaries, potentially creating circuits and logic gates. So this is a really precise method, gives you picoliter accuracy deposition. There's not too much available like this right now from what I understand. So traditionally scientists would do experiments like that by taking one drop of bacteria, or one type of bacteria, and then a different drop of a different type of bacteria, and seeing their interaction. This will allow you to have complex patterns.
At a higher resolution than current integrated circuits are being done?
Not really. I mean, do you mean like traditional, like
Sputtering
mechanical?
techniques and, gaseous distribution and all that stuff.
I'm not really sure about the comparison between, or the resolution that existing electronics have. I can take a look. Yeah.
You know how many bacteria cells are like in one drop?
In one colony. So it's somewhere on the order of 130 picoliter deposition. We haven't gotten to printing bacteria yet, but when we do, we'll definitely look at that. That'll be a good way to explore those methods.
Any other questions? All right, let's thank Steve again.